Hi, today I'm going to talk about my poster, sialidases derived from bacterial vaginosis-associated bacteria and how they impair sperm function by remodeling the glycocalyx. So first, I want to explain what bacterial vaginosis is. It's a dysbiosis of the vaginal microbiome that impacts 29% of women around the world. So in this diagram, a healthy microbiome or homeostasis is depicted with these blue bacillus bacteria that represent lactobacillus, and these are correlated with a relatively quiet immune and metabolic environment and a low risk of adverse health outcomes. Conversely, BV is characterized by a diverse polymicrobial environment where lots of different inflammatory small molecules and cytokines are present, and the pH is much higher, and this is correlated with increased STI acquisition and other reproductive problems like preterm birth and sequelae. So one thing that's really closely correlated with bacterial vaginosis is something called sialidase activity. So I'll get into a little more detail about what exactly sialidases do in a minute, but there was a really cool study done a few years ago where they looked at women with and without BV and characterized how much enzyme activity from these sialidases they could determine. And so in women without BV, the levels are very low, and in women with BV, there's a diverse range, but the level of sialidase activity is even diagnostic. And so the source of these enzymes is actually a couple of different vaginal bacteria. So people have surveyed the genomes of these bacteria and found that a pretty large number of these BV-associated bacteria, their genomes contain sialidase coding genes. Uh, and so these sialidases, what they do is they're bacterial enzymes that cleave cell surface sialic acids. And so sialic acids are glycan moieties that decorate basically all the cells in the body, but especially mucosal cells such as those in the vaginal epithelium and mucins that are both cell associated and soluble. And so here, this diagram shows that these vaginal bacterial sialidases can cleave this purple square, which represents sialic acid. And they may do this for their own metabolic needs or to increase their ability to colonize the host. And so sperm are also cells that are very highly sialated. And so in this study, we look at how removing those sialidases could impair their health by looking at complement activity, sperm's interaction with mucus, and the sperm's propensity to agglutinate, which is a phenomenon where sperms stick to each other. And so the first thing we wanted to do was confirm that these sialidases are actually able to reduce the sialic acid on the surface of the sperm. So we did this by flow cytometry and lectin staining. So we have these fluorescently conjugated lectins, and we can stain the sperm with them and then assess the level of staining by flow. And so in the first graph, we look at mol2, which is a lectin specific to alpha-3 linked sialic acids. And we see that the no treatment control, there's an endogenous level of sialic acid on the cell surface. And when you treat with a commercially available AUS sialidase, that activity is, or that staining is gone. And then you see a similar result with NNH2, which is a sialidase reproduced recombinantly, but is originally from Gardnerella. And we see a very similar result with SNA, which is a lectin specific to alpha-6 linked sialic acid. The graphs look a little different, but that's because of the MAL2 has a secondary amplification step, and the Psi5 is directly conjugated to the SNA, so that's not there for this one. But overall, the results show that BV sialidases are able to remove these sialic acids on the surface of sperm. So our first interesting assay that we wanted to do was looking at complement activity. So we treated the sperm with human serum and then looked at their motility using the CASA, which is a way we can measure sperm into progressively motile, non-progressively motile, and immotile. And so the first thing we did was kind of like a control experiment. We looked at how the sialidase activity alone would impact the motility of the sperm over time. And so the black here is a no treatment control, the green is our positive control commercial sialidase, and the purple is the Gardnerella sialidase. And we see that while the sperm motility does decrease a little bit over time, it does so kind of the same across all the conditions. So the enzyme doesn't impact the motility over time in these shorter time points. Based on that, we went ahead and did our complement immobilization assay where we look at how complement can kill the sperm and that sperm don't move, so they're immotile. Our hypothesis was that removing the sialic acid impairs the sperm's ability to bind complement inhibitors like factor H, and it also may sensitize the sperm to lectin-mediated complement through pathways such as the mannose binding or MBL lectin pathway. And so the first three bars here in gray and white are our controls. So the first is a control where we included the highest dose of enzyme and complement, but we included a human complement inhibitor called compstatin. The second is where we included highest dose of enzyme and heat inactivated complement. And the third is where we included no enzyme. And we saw that the average motility is around 80% for all three of these. And then when we start adding enzyme, we can see decreases in the motility at the highest dose of enzyme, so at 2,000 nanograms per mil, which is the BV-associated dose. We can see that the motility is reduced to about 50%. Next, we wanted to look at cell-cell sticking. So sialic acid is known to play interesting roles in cell repulsion. So removing it might result in the formation of these little cell clumps called agglutinates. And so just zooming in on these little clumps, just to show you how the cells are interacting with each other. So this experiment we treated with sialidase and then some antibodies to see different types of cell-cell sticking. And we have a really interesting assay in our lab where we can time how long this takes. And based on that, we can show it kind of quantitatively. And so here we use three different glycan-binding proteins, so galectin-1, the human contraception antibody, and P-savidum agglutinin, which bind different glycans on the surface of sperm and cause them to agglutinate like the images I showed you before. And so the black here is a control, the heat inactivated enzyme, and the light purple is a medium dose, and the dark purple is a higher dose. And we can see for all three glycan-binding proteins, there's a dose-dependent reduction in the time that it takes for the cells to stick to each other, indicating that the sialic acids do play some sort of role in cell-cell repulsion with sperm. And we hypothesized this might be because the sialic acids are negatively charged, and so they might contribute to this charge-based repulsion. And we confirmed that idea by treating the sperm with the different doses of sialidases and then measuring zeta potential, which is the surface charge of the cells. And 250 and 2,000 nanograms per mil are BV-associated doses, so it was interesting to see that these BV-associated doses are where we start to see the surface charge actually drop. Finally, we looked at sperm transit through cervical mucus. So both sperm and cervical mucus are pretty heavily sialated, and it's thought that that might have something to do with cell-cell interaction and cell interaction with mucus. And so we collected ovulatory human cervical mucin from the clinic and put it in a capillary tube containing a little bit of sialidase at the beginning, and then exposed the capillary tube to sperm and seminal plasma and saw how the sperm are able to swim through the tube over time. So after 90 minutes, we looked at these different distances and counted in the microscope how many sperm were at each time point. And the purple here is the Gardnerella sialidase, and the black is a media control, and we can see that exposure to the sialidase reduces the sperm's ability to swim through the mucus. And this leads us to a couple of other questions that we're now interested in looking at. So we're interested in looking at other glycan moieties because we think these glycan modifying enzymes are a cool tool to study glycan biology on sperm. So now we're looking at other glycan moieties like fucose. We're also interested in how the sperm glycocalyx might impact cell-cell interaction and might impact modified binding to host lectins in the reproductive tract. And we want to expand the study now to additional vaginal bacteria, including Prevotella. And now just a real quick acknowledgements. Thanks to everyone in the Anderson lab. I've had tons and tons of help with this study, including from Europe students listed here. And then we've also had tons of help from the labs next door with reagents and producing proteins, and that was definitely a new area for us, so we got a lot of help. And thanks to the Lewis lab at UCSD, they gave us a plasmid encoding the sialidase that was critical for the study, and then funding sources and the donors that contributed to the study as well.